This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 202, recorded on August 1st, 2019. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, howdy. How you doing?
Last time we saw you in vivo.
Well, I want to thank you so much for doing that interview. You guys, you're amazing. You came all the way from San Francisco just to talk to me. And let me talk about my favorite subject, me. It was a lovely interview. I shared it with family and friends, and it went a long way. So thank you.
I have been wanting to do that for 200 episodes. So I'm glad it all fell together. We were out there. It was number 200. Perfect timing. What do they call it? A perfect storm. Can't get better than that. Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello. What a pleasure to be back with you.
It's been a while since we were here on Skype, but I think over a month. And joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone. And I'd like to confirm that it is always 72 degrees and sunny in San Diego.
It was nice.
It was gorgeous.
Yeah, we had a good time. I just am amazed that it all worked. Everyone's
Yes.
flights were on time, and we had so much extra time built in. It was relaxing. And then, oh, by the way, we went to that restaurant afterwards. Anybody remember the name of it? Rigging, I think it was. Something that had to do with a boat.
Which?
Yes.
Rigging? Hey, Elio, they told us to go.
Sorry.
They told us
about it.
They told us to go because the students said they had the incredible burger. What's the name of the Impossible Burger. Yeah. They didn't. They had a competitor, which is Beyond Burger. And the lady said, "Oh, we used to have Impossible, but this is better."
Oh.
She said it's cheaper and better. So Ray Ortega and I had one. And
I did too.
You did. I didn't think it was so good. But yesterday here on Broadway, there's a restaurant that has Impossible Burgers. And it's called Washington Heights. The restaurant just opened. And that is actually more meat-like than Beyond Burger, I can confirm. Have you ever had one, Julie?
I have. I've had the Impossible Burger, but not the Beyond Burger, so I can't compare.
Do you like the Impossible Burger?
I do. I think it's pretty good, but I also really like bean burgers. So, as long as the texture is there, that's the important thing.
I tell you, if you put the Impossible in front of me and didn't tell me it was that, I would not know. I think.
Who's this Julie that's joined us?
Hi.
Oh, you forgot to introduce Julie.
No, I'm sorry. I'm talking. Also joining us today, we have a guest. She is a science communication specialist at ASM, American Society for Microbiology. Julie Wolf, welcome to TWiM.
Hi, everyone. Very pleased to be here. Oh, of course.
I just kept talking. I wanted to talk about the burgers.
Well, the burger is an interesting thing.
You interviewed Pat Brown, right?
I did because some of the basis of how the components of the burger are made through different microbial processes.
Mm-hmm. And that's on Meet the Microbiologist, which is at asm.org/MTM
for Meet the Microbiologist.
Perfect.
Yep.
And you're right here in New York City, which is why you're here sitting in the office with me.
Yep.
I'm right in front of the wall of polio.
Yeah, and very nice setup for all your podcast recordings.
Yeah, that's why they sound good. Before we get into our science here, if you like our work, consider supporting us financially. Go to microbe.tv/contribute. We have a number of ways you can do that. We'll talk more about it later. Where are you from, Julie?
I grew up in Plymouth, Michigan.
Michigan,
Wow.
yeah. Yeah.
I didn't know that. You knew that, Michele?
Yeah, we've had a chat about Michigan. Plymouth is only, 20 minutes from Ann Arbor.
Yeah, and I went to Ann Arbor for my undergraduate, and I've actually had Michele on Meet the Microbiologist, too, as well as Vincent. So
you went to University of Michigan.
Mm-hmm.
But you
Go
didn't.
Blue.
You didn't have Michele or Kathy Spindler as professors, right? Or teachers.
I do not think I did. No, I remember some of my professors, but I don't think that any of them are active ASM members that I interact with very frequently.
Okay. And I learned that you went to the University of Minnesota.
I did. I went
to do their PhD.
I did my PhD at Minnesota, and that's why I was so excited that Vincent was able to pick up one of these Minfluenza glasses from the ASV conference.
Oh,
which
right.
I was
how
very.
clever.
As soon as I saw it, I thought, I want that. It's very nice addition to
it's
the
a special.
community.
It's a beer glass, right? And Minfluenza, they had a beer made for the meeting.
Yeah, it's a great idea.
Which we've done a few times at ASV, and they called it Minfluenza.
And it looks like it was a wild rice kolsch, which would make sense because wild rice is a very common grain found and grown in Minnesota.
That was one of them. The other was a raspberry something.
Oh, okay.
Which I found too sweet, but I didn't get to have the rice one. But they made a glass for it. And I was setting up for TWiM at ASV, and I got a DM on Twitter from Julie. Can you get me one of those glasses? And I was right across the hall from the registration desk where they sold them. So I immediately went and got one. And so now you have it there.
Yeah, I'm very excited. Thank you.
So enjoy. Don't break it.
No, yeah. Now for the hard part of taking it back to my apartment.
Although they probably have extras, because at the end of the meeting they were saying, "Please, there are more. Buy them." Buy them. And there was also so then Ray Ortega said we should do that at ASM, make a brew.
I love that idea. It's a great combination of microbiology.
Yeah, and you find a local brewery. That's what they did, and they contract them to do it. And in fact, at the previous ASV where they did it, which was in Fort Collins, Colorado, the ASV local host, he went and he added the starter culture, and they, make a ceremony out of it. And he showed pictures of
like
it.
the Olympic torch.
Yes, right, exactly right. It was really good.
Something new for microbe.
Definitely.
The starter culture.
Starter culture, yeah. So I think, if ASM wants to do it in Chicago.
Oh, Lord, there's plenty of breweries in
Plenty
Chicago.
of breweries there, and they should send ASM president.
I believe it's Robin Patel.
Robin
Patel.
Robin Patel,
But
yes.
unfortunately, she's not too far away, right?
Yeah.
Rochester, Minnesota. She could go and put the starter culture in. It would be funny. So anyway, that's the story of why Julie is here. So you did a PhD in Minneapolis on what subject?
I was in their MyCab program, so we studied a little bit of microbiology, immunology, and cancer biology, but my specialty was microbiology and medical mycology in particular.
And then you came to New York for a postdoc, right?
I did. Continuing mycology. I switched from Candida albicans, which I was working on with Dana Davis, to working on Cryptococcus neoformans with Arturo Casadevall.
At Einstein.
At Einstein, Albert Einstein College of Medicine.
We're not too far from here.
Yep. And that's what brought me to New York originally.
Right. But I have to say, it's very hard to get to from here.
I was able to take the subway. I did live in Manhattan and did the reverse commute. And so because it was kind of the reverse, going into the boroughs outside of Manhattan, I actually didn't have a very bad time with the commute.
But from here, it's hard. We usually take taxis because you need to take 6 buses.
Depends on where you live, of course.
And everything. Yeah. And then after that, you went to ASM directly, right?
I did. Yeah. So I started off working a little bit of freelance and then was hired full-time staff in 2015.
Science Communication Specialist, right? That's your title.
Correct.
Right. And you do meet the microbiologist and a few other things as well, right? You're very busy at the annual meeting.
At the annual meeting, that is certainly the pinnacle of our year at ASM. We are very busy. We have a lot of programming that we build into it, and then we execute all of these different programs at Microbe. So it's a lot of fun, to be honest. It's busy, but it's a lot of fun.
But most of the time you're at home.
Yeah, I work remotely, so the headquarters are in DC, but I work most of the time from New York, which is good and bad. It's nice to have colleagues that you interact with, I think.
The water cooler.
Yeah, exactly.
Yeah. Well, they say Twitter is the new water cooler, right?
It is. I spend a lot of time on Twitter, so.
Okay. All right, let's do we have 2 snippets today. When I said we're going to do TWiM on the first, both Michele and Elio had snippets. So I said, well, usually we have a snippet in a paper, but we're going to do 2 snippets. Who's going to go first? I forgot. Is Michele going to go or Elio?
I think Elio.
Elio.
Okay,
I
Elio.
thought I am going to.
Snippetize us.
So the paper I'm talking about is called Gut anatomical properties and microbial functional assembly, promote lignocellulose deconstruction and colony subsistence of a wood-feeding beetle. So this is about the gut of a wood-feeding beetle. It's got 18 authors, all Americans from 11 different institutions. I won't give you all the authors, but the first one is called Ceja Navarro, and the last one is Brody, E.L. Brody. So this is a very thorough study about that sort of summarizes what we want to know about the physiology and anatomy of the wood-feeding beetle. Now, wood-feeding beetles are interesting because they would live on wood. It's not easy to do. They usually consume decaying wood, so the process is already gone a little ways. But what's interesting about them, and it's not surprising perhaps, is that this is a complicated anatomy. A wood beetle is only, a few millimeters, half a centimeter long or something like that. I don't have the exact number. And when you look into it, it has 4 different parts to its gut. 5, actually, if you consider the foregut. The foregut is a little bit like an esophagus, just the tube the food comes in. The food in this case being decaying wood. It goes through a foregut, it goes through a midgut, an anterior hindgut, and a posterior hindgut. Did I say that right? Foregut, midgut, anterior hindgut, posterior hindgut. In each one of those places, something different happens. So in brief, the wood comes in, is chewed up, and it gets depolymerized first in the midgut. That is the first place that it finds, and the midgut is a good-sized organ. So there, the composition takes place. The products of the composition enter the anterior hindgut, and they're fermented there into and things happen there. It's a place where strange phenomena happen, like nitrogen fixation, methanogenesis, and acetogenesis, homoacetogenesis. And then these things go into the posterior hindgut, where supposedly they are absorbed as foodstuff for the beetle. Now, this kind of complicated anatomy is a little bit surprising because if you think about something, polymers get broken down, well, like us, actually that's not so surprising. A lot of the decomposition of proteins takes place in the stomach. Some takes place in the small intestine. Here, what's so surprising is it's not like the rumen of ruminants, because ruminants also eat cellulose-rich material. This is lignin-rich, but there's also a cellulose. And in the rumens, it all takes place in one place. In the ruminants, in the rumen. Here it takes place in 4 different places. So they've worked this out, and this anatomy, I guess some of it was known, some in details probably, some of it was not known. But what they went on to study is the fact that in each one of these compartments, the microbiome is different. Not surprising, perhaps, because different things take place. So and what is interesting is that the midgut, which is where the first decomposition of the lignocellulose takes place, is highly aerobic. And it has a relatively thin wall, which lets oxygen diffuse in, and it is high concentration of oxygen. Depolymerization reactions take place because they're hydrolytic in general. By contrast, the anterior hindgut, which comes next, has a thick wall which reduces the diffusion of oxygen, meaning it's anaerobic, and this facilitates what goes on then in fermentation. Nitrogen fixation, remember nitrogen fixation. The nitrogenase is very sensitive to oxygen and acetogenesis. So this is the anatomy of the beetle. It's an interesting anatomy, but why shouldn't it be? Because it does something interesting. By the way, the result of all this is that not only does it end up with products like glucose, pyruvate, lactase, etc., which the beetle can use. But the fecal material, if you wish, which is called frass, FRASS, is eaten by the offsprings. So the offsprings cannot use wood directly, but they do it because mama eats wood. Okay?
Hey, that's a German word.
Is it?
Frass. Yeah, it kind of fressen means to devour.
Right.
So
And the larvae,
Huh.
the young larvae, don't yet have the chops to eat wood. And so they're completely dependent on this metabolic processing and packaging into frass, high nutrient
Wow.
Right.
particles that the mother produces, or perhaps the father too. I don't know.
I suppose the father does too, yeah.
I
Anyhow.
read that these beetles are very social, that they live in families, multi-generational.
They do.
So
Mhm.
it's very stable social structure.
Right. So let me just tell you a little bit more on what they found. They found that they studied the microbial composition of each department, and they did this in great detail. They sequenced 16 metagenomic libraries and carried out a detailed proteomic and metabolomic analysis. They found 30,000 unique proteins, and about 10% of those seem to be relevant to the degradation of lignin, xylan, and cellulose. And they're also involved in what goes on next, namely fermentation, nitrogen fixation, and acetate production. They reconstructed whole genomes by using shotgun metagenomics, which is no mean trick, and it led to the identification of many relevant microbial species. And these include a number that I had never heard of. Turicibacter and Novosphingobiome. Ever heard of them?
Nope.
No.
Nope.
I haven't either.
No.
So there's new bugs there, which is not unexpected, but the other bugs are sort of well known. So this is the first comprehensive analysis and understanding of the insect digestive system of the Blood Beetle. I thought it was fun. It was plain fun.
It was very impressive. And those microbes that you described are hard at work, because in the introduction of this paper, they point out that these beetles eat 4.5 times their own weight in food each day. So that would be like me eating more than 500 pounds of food each day.
You mean you don't? You don't?
But you don't have to eat wood.
I try my level best, but so those
Okay, try
microbes
not to.
Well, this is why powder post beetles are, if you get a diagnosis of having powder post beetles in your home in the South, it's time to bring out the tent and they begin to fumigate your house.
really?
I mean,
otherwise they'd eat your house up, right?
Well, think about it. Michele just gave us the analogy. She'd have to eat 500 pounds of food, and she's only one individual. These little critters are eating 4 times their body mass in wood, and they are downright scary. And the other fun fact I learned reading about frass, and this absolutely fascinating thing, is if the plant that the beetles are attacking is still alive, the plant cells actually secrete chitinase in response to the beetle's presence, or I should say in response to the frass being present. So frass is actually a signal to the plant. So there may be some microbial signals left behind by the microbes. So if you're looking for a future research project, there may be one out there in frass.
And to just close the loop, the plant excretes chitinases, which then attack the outer shell of the beetle.
Yes.
To fend them off.
Right.
Ah, I see.
Yep.
That's the point.
All right. So, Michael, if your house is infested, you just spray it with chitinase, right?
Yeah, that may be a natural termite and powder post beetle insecticide. Who knows?
So these are called powder post beetles. Is that right?
Yes,
And so they can enter your home because, according to this, they mostly live in the woods, right? But
they enter the woods, but here in the South, we're constantly battling termites, and it has to do with moisture. And here in the damp South, unlike the desert West, we're always inundated with moisture.
Now,
Wow.
now, Elio, the key here is not just that there are microorganisms in the gut that are doing these reactions, but they're different in each stage, right? And that
That's right.
the structure
That's
of the
right.
gut actually reflects what's going on, right?
But look, this is true for us too. We have different bacteria in the mouth, in the stomach, in the small intestine, few, but some, at least the terminal end of it, and the large intestine. So we can brag to be like beetles.
Their figure 6, by the way, is a really beautiful summary schematic that shows the anatomy of the beetle,
right?
And then it shows what metabolic process, digestive process is happening in each compartment, and then it describes which species or genuses of microbes are found in each compartment. So it's a really beautiful summary of an amazing amount of quantitative work.
It is, actually, you're right. I didn't get the whole feel for the paper. It's a big paper. So the 18 authors were busy.
Yeah, I mean, I wanted to comment on that.
They didn't just tack the name on it.
I wanted to say the same thing. They use NMR, nuclear magnetic resonance analysis, all kinds of sophisticated chemical procedures to find out what's going on here, what's being made in the compartments. And I thought it was really interesting. They wanted to look at the transit time of wood from the time they're fed. So they mix it with fluorescent spheres so that they can see when they come out in the frass. So they feed the spheres to the beetles. They starve them first, which I think was funny. You probably have to get Ayacuc approval to do that.
And that may be hard. That may be too harsh.
And then you feed them, and then you watch them, and then that frass comes out, and you look for the fluorescent spheres in the microscope, and then when you see them, then you know that's how long it's taken. I thought that was just great. This is just really a cool study.
It also is interesting to me that they do live in these social communities. So the parent pair, stays with their young until they're adults, which kind of makes sense because the parents are investing all this in generating the food for the offspring, and they don't want interlopers to come and benefit. They want to protect their own genomes. So it's a really interesting biology on many levels.
Yeah, it says they defend their tunnels.
Yeah.
They don't want anyone else coming in. So that figure you pointed out, Michele, I thought it was funny that you start off with what looks like a piece of wood, and then the product is corks.
Yeah.
They draw the frass. I guess this frass is highly structured, right?
Yeah. And it doesn't stick. Yeah. It doesn't stick.
Apparently.
The larvae eat it.
You could imagine that the larvae eating the frass might also be how they start to inoculate their own microbiome so that they can then eat it as adults, right?
For sure. Definitely. Yeah.
Once they get their chops to chew wood. So
in a real way, this is a model system for something that goes on in us. You could say that if you learn a lot about the beetle, you're going to learn a lot about the kind of issues that are involved in the digestive system of humans. I found it's not a big jump.
So this has clearly been done for other organisms, right? Like ruminants, humans,
termites.
So you could imagine that you could do this with a lot of different organisms out there as well. I mean, and you never know when you're going to find one that's really different and tells you new things. So the question is, what else should we do? And we're just limited by money, really, because everyone has a great imagination. And you can imagine doing this with many, many different species if you could, but unfortunately you can't because there's not enough money to do that.
And then you'd want to study it in a cohort that has, for example, Crohn's disease or inflammatory bowel disease and begin to understand pathogenesis and intervention, dietary and probiotic perhaps, or prebiotic interventions. So this is a really exciting field.
And in fact, in Nature Microbiology Reviews this month, August 2019, there's a paper entitled Microbial Genes and Pathways in Inflammatory Bowel Disease. Just anticipating what Michele is suggesting folks run out and do.
Yeah.
So folks are indeed running out and doing the sorts of things that are being informed by this particular type of study. And so when you think about, studying the frass of beetles and how does that affect the human condition, I mean, it's all connected. We're all living life forms, and, the bacteria seem to be in charge of us all.
Are you saying I look like a beetle, Michael?
No. No, I'm telling you, the microbes in your gut are controlling everything.
I agree that this informs human studies, but I also think there's a place to do more of this, right?
Oh, yeah.
For sure.
There are lots of other animals out there and insects which you would want to look at in this way or in other ways, and we can't because much of research is human focused in this country. And, the kind of work that this is going on here is very interesting, but really hard to do because you have to, there aren't that many agencies that will fund it, I guess, is what I'm saying. And that's too bad.
Yeah.
I wish we could do everything we wanted to do, right?
It also would inform different bioenergy projects or bioremediation. If we can harvest microbial communities to break down, let's say, all the waste that we generate in the corn industry,
Right.
all the corn husks, et cetera, and harness that, reuse that energy. Microbes can pave the way.
Yeah, so some of the novel microbes identified here, you might think of using for that purpose, right? Yeah, for sure. That's actually a nice segue to your snippet, isn't it?
Yeah, we're going to stick with the amazing metabolism by microbes with a paper called Draft Genome Sequences of 5 Environmental Bacterial Isolates That Degrade Polyethylene Terephthalate Plastic, better known to most of us as PET. So it's the plastic that water is frequently sold in, for example. This was published in Microbial Resource Announcements, and it is work from a collaboration between people at 2 universities, Willamette University and Reed College, in Salem and Portland, Oregon, respectively. And they are Dr. Rosa Leon-Zayas, Cameron Roberts, Morgan Vague, and Jay Melius. And they are looking to tackle a very large problem, and that is that more than 480 billion plastic bottles are made each year. PET plastic in particular is very convenient. It's inert, it's resilient, it's malleable, so we can use it in many different shapes. The problem is it takes years, if not centuries, to degrade. In fact, our oceans are becoming collection points for all of these waste bottles. So in particular, some of our listeners may have heard of the Great Pacific Garbage Patch. This is a vortex of water that basically has pulled in plastic that can be, for example, discarded on the coast of California, and then years later it will end up in this huge garbage patch, which is now the size of Texas. This is a problem because this debris can hurt the marine life. For example, turtles can mistake bits of plastic for jelly, which they like to eat, or fish eggs, which albatrosses feed to their chick, or mammals can get tangled up in these webs of plastic and drowned. And these huge webs of plastic also block sunlight from reaching plankton and algae, which is a really important food source in the whole marine food web. So it could then indirectly impact many other animals that live in the sea, our food supply, etc. The plastics also leach chemicals and pollutants that can also have harmful effects on marine life and ultimately us. So this team is interested in applying the awesome power of microbial metabolism to address this problem. And their thinking is that perhaps there are microbes that have adapted and have learned or acquired the capacity to break down plastics. And they had reason to be hopeful because there are a couple of reports in the literature. For example, in 2014, a Chinese group described some microbes in the gut of waxworms that can break down plastic. And then there was a 2016 science paper from a group at Kyoto Institute of Technology in Japan that isolated a particular bacterial species that can live on PET as a major food source. So the strategy that this group took was to go to sites that are known to be contaminated with hydrocarbons and collect soil samples. So in particular, one of our undergraduate authors on this paper, this is Morgan Vague. She actually hails from the Houston area. So she went down and traipsed around refineries in that area, or in the Galveston Bay, and collected more than 100 samples of soil, sand, and water, and then took it back to Reed College, where they painstakingly identified different strains of bacteria that were present in those samples, and then asked, do any of these microbes that we got from the oily soil and sand in the Galveston Bay, do they have the capacity to break down oils? So in particular, they did an assay looking for an enzyme called lipase. And the way they did that was they made agar plates that had olive oil as the primary food source, and it had an indicator dye, which is called
rhodamine B.
There it is, rhodamine B. And rhodamine B can bind to fatty acids that get liberated from when hydrocarbons are broken down, and then it will fluoresce if you shine a UV light.
Michele, I have to say that using olive oil as a sole source is perfectly fine with me.
It's very sophisticated. Yeah.
Speaking of the Mediterranean diet.
I don't know if it was virgin olive oil or first press, second press.
First press, right?
Was it Italian or Spanish? That's the big
debate.
I
don't know. But they found 192 different microbes that had some evidence of lipase activity, and 3 had quite a bit. So now they subjected these
microbes. By the way,
microbes.
this is really ingenious. It bears saying that this is an ingenious way for looking for plastic degraders by looking for bugs that have lipases. I wouldn't have thought of that.
And this is an undergraduate senior thesis project, which even makes it more remarkable, Dr. Schaechter.
Yeah. Yeah, and I'd like to talk more about the individuals too, but let me go through with the next steps of the science. But I would definitely like to highlight this as really impressive work at primarily undergraduate institutions, Reed College and Willamette University. So once they had candidates that were growing on the olive oil, they then did a more stringent test. They asked if any of these 192 different strains could grow on this PET, a plastic that's common in our water bottles and other disposable bottles, and that's now accumulating in these massive acres of trash in the ocean. So to do that, they first pre-treated the PET with UV light, because they knew that would break down some of the polymers and make the PET more easy to digest. They then set up cultures, bacterial cultures, that grew at 30 degrees with some shaking for 6 weeks. And then they looked to see if the amount of granular PET they put in at the start was the same or if it had decreased. And their data is shown in Figure 1, where they were able to measure a modest but yet clearly reproducible decrease in the PET granules. And for example, one of the consortium, so they ended up with, from the colonies that were had lipase activity, they ended up with 5 different samples. Consortium 9 was 2 different strains of bacteria. Consortium 13 was 2 different strains. And then they had another strain that they tested by itself. So they tested each of those.
This is also remarkable that normally when you isolate bacteria, you isolate single colonies or single species. Here you isolate consortia.
Mm-hmm.
Yeah, from a single colony, they found multiple strains. So those bacteria must adhere to one another in order to not be purified by our standard colony purification strategy. Is that what you're thinking?
Yes.
Yeah. So they tested each of these consortia independently, and then they combined them as well to do this quantitative test and look if there was degradation of PET. And one of the methods that they used to measure it was, again, a very sophisticated nuclear magnetic spectroscopy. And Cam Roberts, one of the other undergraduates from Reed who worked on this project with Morgan, wrote that she was so excited during this assay. She had lots of different samples to test. She was looking for byproducts of the biodegradation of the PET. And was just so eager to learn the results that rather than just using the automated spec where she could have set up the samples, gone home, and then looked at the readout the next morning, instead she sat for 2 hours as each sample went through and the numbers came up on the monitor. And she said she found herself just squealing as each new sample finished because she saw that eureka, there was degradation. And she said that moment has really stuck with her and taught her how much she loved research. So having identified some activity of interest in these samples, they then took another sophisticated approach. They did whole genome sequencing of each of the 5 microbial strains using Illuminamyces. And in Table 1, they show the results there, all their statistics for doing the fancy whole genomic sequences. But they also show that they isolated 2 different Bacillus strains and 3 different Pseudomonas strains. And they describe kind of the most likely species that those members would belong to. So the next bioinformatic approach that they took was to try to get some insight into how these microbes might be attacking the PET and using it as their sole carbon source. So there they used a software program called KEGG, which some of our listeners may have heard of. It's a algorithm that can predict metabolic pathways from whole genome sequence data. And sure enough, they did find a number of lipase-like enzymes and also enzymes that are part of other metabolic and biosynthetic pathways, nitrate reduction, et cetera. So now they're in a position to continue this collaboration between the group at Willamette University and Reed College. They've also recruited a chemist from Reed College to try to then to identify the molecular and biochemical pathways that are dedicated to breaking down the plastic. And for that, they've applied for funding from the National Science Foundation that is specifically designed to support hands-on, sophisticated research projects at primarily undergraduate institutions, which is where this work was done. So let me just say a few things about Reed College. It is a liberal arts and science college in Portland, Oregon. There are about 1,400 students there, and they really pride themselves on recruiting and developing students who exhibit engaged citizenship. And the same is true at Willamette University. That's got about 2,000 students, including some at a law school and a school of management. But they too pride themselves on civic engagement. So it's clear that the undergraduates, Megan and Cam Roberts, are perfect examples, and I'm sure they have brought a lot of pride to their university in carrying out this project. I'll also add that their advisors are to be credited for taking this bold approach. Jay, I've known for a number of years through the pathogenesis field, says that he really had to step out of his comfort zone because this is not what he normally studies. But it's been great because he and the students have been learning together through the process. And then he is fortunate to be collaborating with Rosa Leon-Zayas at Willamette College. She's an assistant professor of biology who's an environmental microbiologist and bioinformatician. She hails from Puerto Rico, where she got her bachelor's degree, and she is continuing to focus her energies on making sure that underrepresented minorities in the sciences, especially Puerto Ricans, Hispanics, and members of the queer community, have opportunity to learn firsthand the excitement of science. So
she traveled via San Diego, and I got to know her there.
Oh, did you? That's great. Yeah, she she studied ultra deep sea single cell microbes from the Puerto Rico Trench at Scripps Institute of Oceanography. That's neat. Right. And then did her postdoc work at University of Delaware. So that is the story, and I'd really am delighted that these two undergraduates got to really contribute firsthand to a cutting-edge research project. Cam Roberts was one of the undergraduates who did this work, and her colleague Morgan said that it was really Cam that showed amazing creativity coming up with a process to analyze plastic degradation. Wow. And Cam is now a research assistant at Oregon Health and Science University in Portland, Oregon. And Morgan aspires to enter graduate school or medical school in the future. So hopefully through this project, they've gotten hooked, and we will continue to hear great things from each of them in the years ahead.
I see Morgan has a TED Talk on this as well, right?
She does. Yeah, and she actually won a prize for her thesis work, too, the Class of 21 Award, recognizing the creative work of notable character involving an unusual degree of initiative and spontaneity.
And Julie, this was featured also somewhere on ASM, right?
Yeah, so the new microbiology resource announcements is kind of a transition from the old genome announcements. It used to be just for genomes, and now it's for all types of different resources, including consortia of genomes, like in this paper. And every month, I work with the editor-in-chief, Irene Newton, to select the microbial resource of the month. And this was just featured last month, I believe, in June as our microbial resource of the month, where the researchers themselves answer a series of questions about what their resource is. And in this case, it's the 5 genomes. And they are able to provide some of the basic questions where they were doing this research, what they think the resource will be used for in the future. And on that page, and I can send you a link, Vincent, we have pictures of Cameron and a link to that TED Talk that you guys mentioned.
That'd be cool.
Just another comment about microbial resource announcement, this journal from ASM. It's online only, and it's fully open access. And it is a place where authors and practitioners can go for big data sets, metabolomics data, proteome data, also information about culture collections or different mutant libraries. So in other words, let's share the resources that we've developed and put them out there for the community to really accelerate our interdisciplinary research across the field. So exactly. I think it's a really great new journal, and I thank Irene Newton for her leadership as the editor-in-chief.
And if you're into looking for resources for your high school students or any student who's curious, I just want to commend ASM, and I don't want to embarrass our guests, but I think the way they present the data on this microbiology resource site is really quite compelling. It's very easy to read. It's got great pictures. There's a chemical structure of what the polyethylene terephthalate looks like, or PET, how it looks like in the garbage patch with all these plastic water bottles compressed. And then it goes through a question of answer. And you can actually see Cameron Roberts at the bench enriching the bacterial consortia. So you too can do this, and this would be a great resource for our high school teachers who use TWiM to encourage their students about thinking about science projects or anything to do with science. It shows them all things are possible.
I'd just like to echo what Michele and Elio have already said about the power of using this type of search for different types of plastics, because PET is just one of the many polymers that are contaminating our waterways and our in this case, I think they were working in a Superfund site, which is a site that is so contaminated that the EPA has designated that it deserves some federal funds to help clean it up. And you imagine that because of the quick generation time of microbes relative to people, that perhaps there are microbes that have learned how to degrade other polymers. And this type of research is really applicable for a number of different potential contaminants and an answer for cleaning up potentially some of these Superfund sites.
Let me make a different point here. In a way, it's surprising that being that there are bacteria which can eat plastic that are not more oftenly found, that is, they don't do an awful lot of chewing. If you were to contrast this with isolating bacteria which degrade polymers from soil, you would find a tremendous amount of activity. You would take some soil, you would put it on a petri dish that has some agar, and and the only source of carbon would be the polymer that you designate, you'd find a tremendous amount of stuff. And when you look in the soil, it would be there. In the ocean, that's not the case. Why is it that these fantastic bacteria aren't already doing the job? And I don't think we know the answer.
The answer is dilution. I mean, it's in the ocean.
Well, so what? And it's not dilution, because the plastic is not diluted. It's a piece of plastic.
Yeah, it needs a nitrogen
I don't think
source.
that's the answer at all.
Well, one could think that perhaps the selection force hasn't been around long enough for them to become a major population. We don't know what's out there, of course, but the plastic hasn't been there for, I don't know, maybe more than 1 or 2 human generations.
Yeah.
And a clue from this work, too, is they found that they had to pre-treat their PET with UV light in order for the microbes to be able to use it as a food source. So perhaps once the bottles get into the ocean, they're deep enough in the ocean that they're not getting enough UV. Maybe that's one way we could think about accelerating the process.
It serves to break that aromaticity of the complex aromatics of the terphthalate.
I think the point is that finding the bacteria is step 1.
Absolutely.
Absolutely.
Step 2 and 3 have to come.
Yeah, so that's what I wanted to ask Michele. What is the next step? It's certainly not using these isolates to degrade plastic, right? What's going to happen next?
So it's a good question. I think one thing that should happen is we should get more people working on this problem, because as Julie mentioned, it's a diverse problem. It's a huge problem, and we can't leave it up to inspired undergraduates at primarily undergraduate research institutions to solve this major world problem. So I'm hopeful that more attention will be paid. And then in terms of the science, boy, I don't know. I sure think that we need to not look for one bug that's going to do all the work, but start to use more sophisticated methods to figure out which population, food chain, much like Elio taught us in the wood beetle. It's going to take a complex community to do this efficiently, and hopefully eat 4.5 times their weight in plastic each day.
Amen. Yeah.
Well, the headline in this morning's paper is Charleston Plastic Company cited for pollution after a nurdle spill on Sullivan's Island. Nurdles are the precursors that they use. These are these little tiny plastic pellets. And Frontier Logistics was cited for violating the state's Pollution Control Act after it found massive amount of these loose plastic nurdles at the company's shipping facility at the Charleston port that washed up on Sullivan's Island's beach. And so these are really, really small, little tiny plastic pellets. These aren't, your water bottle. They will become your water bottle. And they are sitting in the Charleston Harbor right now, and little shrimp and fish are eating these things.
What a shame.
This is not just out in some mysterious place in the Pacific Ocean in the garbage patch. This is in my backyard this week.
Yeah.
So let me end on an optimistic note here. Morgan Vague came to science by a non-traditional route. She had been a student at Houston Community College and then came to Reed College at age 22. She is now the holder of a U.S. patent that is titled Bacterial Compositions and Methods of Polymer Degradation Using the Same, thanks to really the inspiration that both Morgan and Cam said they got from Jay Melly's undergraduate microbiology course. So hats off to everyone out there teaching our undergraduates and our high school students about biology. Projects like this can really inspire people, and that we need all the minds we can get working on these big problems.
Yeah, it's great. I love it. I have to say that I have to tell you a very short story. Many years ago, I was actually at ASM headquarters in D.C. I don't remember why, but I went out to lunch and was walking in not towards Connecticut, but the opposite direction. And there was a building with a huge poster on the side, which was the cover of National Geographic where they had an iceberg, and then below the ocean was the rest of it. It was a garbage bag. Do you remember that image? And it was called Planet or Plastic, right? And it is iconic. It just looks like an iceberg, but then it's got the garbage bag below with the two handles. It's just beautiful. I looked at it. I'd never seen that before, and that was during a visit to ASM. So talking about the plastic problem, that was an entire issue devoted to it. Really cool. So
let's find that. Let's find that and add it to the show notes. That's great.
It's a very cool. I got it right here and I'm looking at it, and I will do that. It's really cool. All right. I want to let's do a couple of emails here at the end, and all three of them are related to episode 200.
Our field trip.
The first one is from Stan Malloy, who writes, "Hi, TWiM team. We thoroughly enjoyed your visit to SDSU. Thought you might enjoy this fun, short, insightful JMBE article by Elio on a graduate integrative microbiology course he's been co-teaching. In case you'd like to link it as a follow-up to his interview, the article is open access." And this is another ASM publication. We figured it out. It's an article on your course, Elio.
Well, I'm glad he points to that. Very nice one.
So this is interesting because it's a course where you get speakers in every week or two, right, to talk about their work. And then
every lecture
is a guest, right?
That's right.
And then I like the final exam. Write an essay on a topic of your choice.
The easiest question to write, the hardest to answer.
Well, I can imagine students saying, "What am I going to write about?"
Well, it's work for both the student and the poor soul faculty that has to grade it objectively.
It's fun, let me tell you. It's pure fun to read them.
Elio, the problem is, this is a great idea. I love it. Getting a speaker every week or other week, who pays for it? If I propose that here,
pays for it.
Are they local?
Are they all local?
Lecture.
They're all local then.
They're local guys.
Okay, so that's the thing. You have to have, 12 or 15 local people that can do this, right?
What's the problem? You don't have that in New York City?
No, we don't have that.
I didn't know it was that poor in talent.
Well, it depends how you want to organize the course, of course, right?
Exactly. You organize it any way you want.
Yeah. Well, first of all, I thought it was people coming from a distance, which is a real problem because we have no money for that. And if I co-opted my seminar series for the department, they would say, "No, you can't take these people and teach a course with them." But if
no, none of that.
So you get all local people.
Yeah, and they love coming to give this lecture. It's amazing. I never had anybody say no to us.
I like the way you ask them to talk, right? Let me see if I find it here. You tell them, 40 minutes to tell the students that theirs is the finest field in all of biology. Isn't that great?
Why?
I like that too.
I like it. That's the way you get people to do it.
Yeah.
Anyway, I'm going to try and do a course like this and see if it works here at Columbia.
It does. Let me tell you.
Well, I may not organize it properly. That could be the problem, right?
It could be.
But I would like to do it because I do love teaching. All right. Another email from Sue Ellen. OMG. Just listened to the TWiM featuring a stroll down memory lane with Elio, and it was awesome. He really is a remarkable person and scientist. I loved his stories. Science needs more Elios.
Here.
I agree.
Yeah.
To say Sue Ellen.
What can I say?
Sue Ellen is a regular listener of TWiP. She did a little crossover here, Elio. All right, then the last one is from Darak. He writes, "What? Elio, you're that Schaechter? Elio is short for Moselio?"
Yes, it is.
The back-to-back papers that you, Kelgard, and Malo did in 58 were so cool. I was really interested by the shift-up experiments because my dissertation was about mRNA regulation after an upshift in yeast. Steady-state physiology at different growth rates is, of course, foundational to this process. I once heard someone say, systems biology is just physiology with marketing. I love that. And I think that microbial physiology lives on inside the field of systems biology. Even now, the field is working to describe the mechanisms of the enteria of growth rate described by Arthur Henrici back around the time when Elio was born, I estimate.
Yes, that's right.
Yeah, you know that name?
Oh, yes. My hero.
Elio, your 2015 review points towards Terence Wa's group's work modeling the balance of gene expression at different growth rates. And I figured I'd mention two other extensions of these ideas. Nama Barkai's group has been doing awesome work in yeast recently in 2016 Cell and 2017 eLife, examining the dynamic limitations of proteome allocation in different growth conditions and transitions. Great work. On the modeling front, Giordano et al. And PLOS Computational Biology 2016 demonstrates in silico an intriguing strategy to the problem of resuming growth rapidly, the bang-bang singular. Maybe the physiology of microbial growth would make for good TWiM. Best of luck, Darak. You don't know Darak, do you, Elio?
Well, say that again?
Darach. That's the first name.
Uh-huh. No, I don't know her.
Well, they know you.
But I'm very grateful.
All right.
Thank you.
You're welcome. And I just want to do two more quick ones here. One is from Mark. You may have already seen this New York Times article on vaccination safety. Vaccine injury claims are few and far between. Really good article. Perhaps citing vaccination safety facts similar to these would be a good complement to your passion regarding the issue. So I'm often talking about vaccination and the need to do it. And, you know, people who are against vaccines often say, well, they have side effects and they're so serious. And in fact, this article shows that the vast majority of side effects associated with vaccination are relatively minor, and fever, pain at the injection site, and so forth. And so I think that a lot of the fear is unfounded.
And the trade-off is certainly worth it when you're talking about diseases that can cause, death and a lot of lost days.
For sure. So this is a Times article. We'll put a link in the show notes. And then we have another one from Mark, maybe the same Mark, and he sends a letter, a link to an article. Programmable bacteria induce durable tumor regression and systemic anti-tumor immunity. Sounds like a very interesting paper. I wonder if you could review it on TWiM. By the way, I'm not sure if it's my exposure to your TWiM, TWiVO, and immunology podcasts or yours, but I really like the way you seem to be increasingly discussing the overlap between these fields. It's great. Thanks, Mark in Missoula. Well, Mark, I have a little treat for you. This paper, it's an amazing paper, so cool, just published in Nature Medicine. You inject mice with bacteria, E. coli, and they home to the tumor. So you can load the bacteria with things that will kill the tumor. And it works.
Is it antigens? Do the bacteria express antigens that are present on the tumor cells?
So in this case, Michele, they used an antibody to CD47, which normally is an antiphagocytic receptor on the tumor surface. So you block it with this antibody, and the phagocytes come and kill the tumor cells.
And eat the tumor cells. Wow.
And so this turns out to be a protein on the surface of many tumor cells. So they engineered it in E. coli. They used nanobodies, single-chain antibodies. The cool thing, though, is when you inject mice with E. coli, first of all, they're fine, which blows me away, intravenously. And secondly, they home to the tumor specifically. They go in, they make this antibody, and the tumors are completely eliminated. It is a killer. In fact, there's a guy at San Diego, Jeff Hasty, who pioneered this idea that the bacteria are engineered to go through cycles of growth and lysis using quorum sensing. Just amazing. And one of the authors here is a postdoc, former postdoc, and they engineered it to do this. Now, I have to say, the authors are here at Columbia University.
We should bring him on.
I did, so I brought him on. It's already in the can, coming out in September.
It's already in the can?
For your immunology podcast, or is that?
No, it's going to be a TWiM. I did it for a TWiM, and I grabbed them one day, and I recorded it. So Nick Arpaia and Tal Danino. Nick is an immunologist here in the medical school. Tal is downtown. and they talked about this. So, Mark, you say jump, we say how high.
How high?
There you go.
That's a great way to close.
All right, so that is TWiM 202. If you listen on a phone or tablet podcast player, please subscribe so that we know how many people are listening. And if you really like what we do, please consider supporting us financially. You can go to microbe.tv/contribute. You could donate, say, $1 a month on Patreon or PayPal, or you could buy T-shirts or mugs. We would love that because we have expenses, and also we'd love to be able to travel and take the team with us to remote sites and bring you cool science from all over the world. So microbe.tv/contribute. And, of course, we love getting your questions and comments. TWiM@microbe.tv. Our guest today from ASM here in New York City, Julie Wolfe. Thanks, Julie.
Oh, thanks so much for the opportunity.
Where can people find you?
They can find the podcast that I host at asm.org/MTM for Meet the Microbiologist. And I am also on Twitter at Julie Marie Wolfe.
All right, great. Michele Swanson's at the University of Michigan in Ann Arbor. Thank you, Michele.
My pleasure.
Elio Schaechter is at the wonderful blog Small Things Considered. Thanks, Elio.
My pleasure, too, indeed.
And Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank ASM for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.